Good morning, everyone. I would like to thank for the IWCL committee for selecting my abstract for oral presentation. I have nothing to disclose. So worldwide sequencing efforts have consistently identified RNA splicing factor SF3B1 as one of the most recurrent mutations in CLL. So this gene mutation appears to co-occur with mutation in ATM and deletion of 11q, which the minimal deleted region contains ATM. So the causative effect of SF3B1 mutation, either by itself or together with ATM deletion, remains elusive. So we generated the animal murine model that conditionally expressed heterozygous SF3B1 mutation in B cells. Analysis of this animal model indicating that expression of this heterozygous mutation leads the B cell to undergo cellular senescence, while co-expression of ATM deletion override the cellular senescence and result in low penetrant CL in aged mice. So we consider this model as a very nice model to further study the fundamental molecular and the underlying mechanism of how SBM mutation together with ATM deletion leads to CLL. So we first performed the RNA splicing analysis in the spindle B cells which harbor different deletion and mutations. Our analysis indicating that so both SBM mutation, ATM deletion, they all have alternative RNA splicing. And I think the most important thing to point out is all these spec variants impact the DNA damage pathways. So to further understand how these spec variants associated DNA damage response impact DNA damage, so we first checked the gamma H2AX, an indicator for the genomic instability in the spindle B cells, which either have the SBM mutation or ATM mutation or the double mutation. So clearly here we see that SBM mutation has higher level of gamma H2AX signal, while double mutant and double mutant without CL all have higher level of gamma H2AX signal, suggesting there is a synergy between the SBM mutation and ATM deletions. We further confirm using the COMET assay that SF3B1 does induce double-strand break. So this raises up the one question: what is the role of SF3B1 mutation in the regulation of genomic instabilities? So we consider whether or not R-loop accumulation can be one potential mechanism. So R-loop is a link between the transcription and the DNA damage response. So we know that RNA splicing is transcription coupled. This is a natural product, contains 3-strand structure which have the DNA RNA hybrid plus the single-stranded DNA. So normally as a natural product, but when it's accumulated, it can generate replication stress and genomic instabilities. So in the past 2 years, RNA splicing factor mutation in U2F1, SR, SF2, all indicating that they are associated with increased R-loop formations. So this structure can be detected by a specific antibody S9.6 by either immunoblot or the immunofluorescence. So in addition, this structure can be degraded by the RSH1. So my first question is whether or not we can detect accumulation in the SBM mutant cells. Our immunofluorescence staining indicating that there is accumulated S9.6 signal within the nucleus in the mutant cells. We actually further confirm this observation in the isogenic cell line which harbor the heterozygous SBM mutation at endogenous loci. We further show that when we overexpress RSH1, the S9.6 signal is significantly reduced. Using another orthogonal method, we further validated that we can see increased R-loop formations in the SBM mutant cells, and they are very sensitive to the RSH1 treatment. So our next question is, if we remove R-loop, whether or not we can reduce the DNA damage. So using the num6 cell line, we overexpress either control vector or the RNA-H1, and then we measure the DNA damage using the commit assay. So clearly we see that overexpression of RNA-H1 removed R loop and then reduced the DNA damage. So in conclusion, we show that SF3B1 mutation induced R loop accumulation and double-strand breaks. Removal of R loop reduced the level of SF3B1 mutation-associated double-strand breaks. So with this, I would like to thank my group members, especially Martina Susan. So she has been performing all the DNA damage-related work. I would like to also take this opportunity to thank my previous mentor, Catherine Wu, and my current mentor, Dr. Steve Rosen, for their continued support. Also my collaborators within, outside of City of Hope. One more thing is, my group is currently recruiting postdoc fellows. If you have interest, please come visit my poster. Thank you.